Hey, hello everybody. Welcome to the bio panel discussion on the role of new technologies in cell and gene therapy. My name is Doug Miller. I'm a managing director at the Bioprocess Technology Group within BDO. We provide CMC consulting services to companies in the biotech or biopharmaceutical industry, helping them do their products. And we focus a lot on the cell and gene therapy space as well. And I'm going to be the moderator for the panel today. And we have an esteemed group of panelists. We have Daniela Kranchik from the Dynamic Capital Group. We have Tim Kelly, President of Manufacturing at Askbio. Andy Ramelmeyer, Executive Vice President, Technical Operations at Sangoma Therapeutics, and Ran Zhang, Chief Technical Officer at Orchard Therapeutics. And I'm really excited to hear everyone's thoughts on how technology is supporting the development of cell and gene therapy products, and what types of technologies may be needed in the future to really help facilitate the development of these products. I'd like each panelist to give a short introduction of themselves and their company and their focus, starting with Daniela.
Thanks very much, Doug. Appreciate it, and thank you for the invitation to be here today. Daniela Kranjak, I'm founding partner of Dynamic Capital. We are a growth equity and venture capital fund. We're focused entirely on life science industrials. It's a bit of an interesting play, filling a much needed gap in the funding landscape. We focus entirely on the tools, technologies, and services that are really fundamental for biotech and pharma. That addresses basically the full range of biotherapeutics, from mAbs all the way through, of course, to cell and gene therapy. It's a key area of growth and opportunity for us and our fund. We invest, just to give the audience a sense, we invest in companies basically at the point of proof of concept, early revenues or revenues and looking to scale. We've invested in companies such as Rooster Bio, Selfie Biotech, and Envisagenics, among others. And we really created Dynamic out of a need from our experiences as entrepreneurs and as acquirers in the space at companies like GE, Cytiva, Sartorius, and AGC Biologics. So we really saw that gap in the funding landscape for novel tools and technologies and services in biopharma underpinning those innovations. And we wanted to create a fund that would be not just capital, but the supporting structure that these typical technical founders need.
Oh, thank you, Danielle, for that introduction. Thanks. Tim.
Yes, thanks, Doug. And again, thanks for inviting me to participate. I'm Tim Kelly. I'm president of the manufacturing business unit for Askbio's biopharmaceutical, more commonly known as Askbio. Askbio is an AAV-based gene therapy company, clinical stage gene therapy company. We have 2 programs in the clinic at the moment. And really, Askbio has always been focused on manufacturing of these technologies. Our scientific founder and chief scientific officer, Jude Samolsky, is a fairly well-known figure, a pioneer in AAV biology. And I think Jude has always been very focused on accessibility of these therapies. And that's really translated into our efforts to develop manufacturing platforms to enable sort of cost-effective production of these therapies. And so, we've been able to sort of develop over the years a proprietary cell line and manufacturing process for AAV-based therapies that's sort of serotype agnostic. And we've actually implemented that platform in a variety of different settings. We've been able to tech transfer it a couple of times, including some, I think pretty well-known acquisitions, such as the Pfizer's acquisition of Bamboo Therapeutics. And today we have a couple of joint venture partners to deliver these technologies to the marketplace. So we have a joint venture called Viralgen in San Sebastian, Spain, which is focused on AAV production, as well as a joint venture called Touchlight AAV, which is focused on the production of alternative technologies to plasmid DNA for the production of AAV. So thank you again for inviting me to join.
Yeah, it's great to have you on the panel, and look forward to hearing your thoughts on some of the technologies that we're going to be discussing. Andy, would you please introduce yourself and Sangoma?
Yeah, thanks, Doug. And again, thanks for having me and spending the time together to talk about these relevant issues. I am head of technical operations at Sangoma Therapeutics. Sangoma is probably one of the oldest of the companies in the at least in the gene editing and gene therapy as well, and cell therapy business. They've been at it 20 years with their zinc finger technology. It's not been until the last 5 years we've been trying to translate that zinc finger technology into therapies. Now, we have, with gene editing, there's work to be done to make that clinically meaningful, and that's a focus of our business. In the meantime, we have a number of products in the gene therapy space that aren't necessarily focused on gene editing in both hemophilia A, Fabry, PKU, and a host of other liver-focused, as well as in CNS, which is a partnership that we've done recently with Biogen. And that one is in a different focus where we're looking at gene, not editing, but gene regulation. And if anybody that understands what zinc fingers do, they are natural in the body, and they help regulate at the DNA level, how the DNA modulates, manufacture certain proteins, like proteins for tau that are impacting in Alzheimer's and the like. So we are looking at using our tools to make genomic medicines. Now, we also are focused in cell therapy. We've had a number of programs already underway in collaboration with Sanofi on beta thalassemia and sickle, and also with Kite on making an allogeneic T cell. We are expanding in another area, which we're one of the first in regulator T cells through our acquisition of TXL back in 2018. So we've trying to establish ourselves broadly in a way that allows us to manage our risk through these various programs. Gene therapy, which we think is low-hanging fruit, is the first wave of our products. We have investments in cell therapy, and as I mentioned, gene regulation with our partnership with Biogen, and then eventually gene editing, which we see as the holy grail of this business, to go into and actually modify the gene at the DNA level and to change the course of disease. So I'm looking forward to the ongoing discussion, and obviously my time at Sangoma has been it's been a handful as we've been managing a lot of these different programs and trying to rationalize and focus them as best we can.
Yeah, thanks, Andy. Thanks for being on the panel. It's certainly impressive the range of different plays that Sangoma has in the cell and gene therapy space, and yeah, looking forward to hearing your thoughts on what some of the key challenges that you're facing are. So thanks. Hey, Ran, please introduce yourself and Orchard.
Sure. Thank you, Doug. Thank you for inviting me to the panel. I am Ran Zhen, Chief Technical Officer at Orchard Therapeutics. Orchard is a gene therapy company primarily targeting rare genetic disorders at this stage. We use gene-modified hematopoietic stem cells to introduce the correct genes to patients who have the missing genes or the faulty genes that are required for normal physiological functions. The HSC-based gene therapies offer great potential to treat a wide range of genetic disorders, such as neurometabolic disorders, primary immune deficiencies, blood lineage disorders. Because the correct genes are introduced into the stem cells, these therapies have demonstrated durability and considered once and done cures. And we have patients with the longest follow-up up to 18 years and phenomenal safety profile and clinical efficacy. Currently, Orchard has one product on the market for ADHD, the so-called bubble boy syndrome, and the 2 late-stage products and a number of early clinical products. The one for metachromatic leukodystrophy, neurometabolic disorder is currently under the review with EMA. And really glad to be here and love to hear the thoughts from the fellow panelists and from you, Doug.
Yeah, thanks, Rand, for being part of the panel. And yeah, they're really looking forward to hearing your thoughts about especially the challenges in the ex vivo gene therapy space. So welcome. Well, great. Thanks all for the introductions. I'm going to pose a first question to you, Tim, and it's sort of a 3-part question. And it is, first of all, what do you see as the most significant challenges in the development and manufacturing of in vivo gene therapy products, such as those that Askbio is working on? And then what are sort of the current state-of-the-art technological solutions to those challenges? And then what do you see as sort of the needs for future improvements in technologies to really, really enable this field to develop and to be able to manufacture these therapies in a cost-effective, efficient, consistent way that can enable the economics to work out for the therapies as well?
Right, thanks, Doug. So yeah, it's a complicated topic. So I'll do my best to touch on as many of those kind of aspects as I can. Certainly, I think when we talk about production of viral vector, kind of overall productivity and yield, is still a challenge, right? And obviously, this field started primarily using adherent cell lines and growing up cells in cell stacks and so forth. And for really low dose products, eye and ear indications and the like, that actually is still really a viable manufacturing platform, as inconvenient as it may be from a processing perspective. Many companies, including mine, Askbio, have transitioned to suspension cell cultures and able to achieve much more economy of scale there, but still generating sort of sufficient quantities of vector to yield kind of the dosing requirements, particularly for systemic administration, is definitely still a big challenge. And this is where I think we focused a lot of energy over the last maybe 8 or 10 years to develop proprietary cell line and production process to try and address these challenges. So we're able, I think, still within kind of your conventional transient transfection mode to enjoy relatively high yields compared to what's published elsewhere. But that still comes with a host of challenges. Plasmid supply is a challenge, right? It's a critical raw material that is high cost and takes a long time to produce in conventional E. coli fermentation, production. So that's a big, kind of area for improvement. Further optimization of these, suspension-based transient systems and scale up. I think there's always been some, kind of doubt as to how scalable these processes can be. And certainly, compared to a stable producer cell line, there's going to be some limitations with production scales. But we've been able to scale our process up to 2,000 liters now, and kind of given the productivity, we believe that we can address kind of the commercial market demand for a pretty broad range, certainly of sort of the monogenic types of diseases, even where systemic administration is required. But obviously that's just the first frontier, and as we think about pathway diseases and more complex types of applications, there's going to be a need to go further and do better there. So I think in terms of technology development, again, the stable producer cell line is definitely kind of the goal, I think, for a lot of companies, including ourselves. We're actively working on that, and the team from Symphomics that we brought into our business last year, who are experts in regulation of gene expression, are helping us to try and solve that problem. Again, also working on the plasmid challenge with the transient applications through our joint venture in Spain called Touchlight AAV. So developing technologies to provide more cost-effective and less time-consuming ways to generate transfection starting materials through the doggy bone DNA, as it's popularly known. And then another area that I want to touch on for this question is analytical characterization. I think today we're still in certainly at least in the AAV space, we're relying primarily on kind of pretty old school analytical technologies. We're actively working on trying to bring our programs kind of into the 21st century with conventional analytical techniques that are commonly used for therapeutic proteins, for example, using more HPLC and CE and mass spectrometry techniques, so we can really understand the potency and further optimize kind of our ability to predict dosing and so forth, and really get to more of a well-characterized product. I think today in the viral vector space, for someone like myself who spent most of my career in the therapeutic protein space, it's tough to consider these as well-characterized biologics, compared to what we're able to do with proteins. So that's certainly an area that we're focused on, and that I think is going to need to be a focus for our industry going forward.
Thank you, Tim. Wow, yeah, a lot of different approaches that you guys are taking to make these products a reality. Very interesting. Andy, I'd like to pose the same question to you, since Sangoma is working on a number of in vivo gene therapy products, as you mentioned. What do you see as some of the biggest challenges facing Sangoma in the development and manufacturing of these therapies, and where do you see technology currently being employed effectively to address some of the solutions, but also, where are the gaps? Where do you think that the technology ultimately needs to go to get, these products to the market effectively?
Yeah, so we're one of the early adopters of the SF9 insect cell culture platform, and we saw the benefits of that early, in that we have yield and productivity issues that were just discussed, and that's something that we're a bit of the Wild West right now if you think about where we are from that end. We also rely on a transient infection process, which if you just look at monoclonal antibodies, if that's our baseline, which they're very mature in their processes, if we want to move to that direction, and it would require thinking about a producer cell line. And there are a number of companies that are developing technology in that field. There's also a number of innovator companies that are developing their own producer approaches. And the advantage there, though, is it will allow you to move away from having the multiple pieces that need to be managed. The generating for SF9, you have to develop what we call bacillo-infected insect cell banks. It's called a BIC. And that is needed for each process. And it needs to be done, if you start thinking commercially about those BICs, those are like our research master cell bank transition. And the issue is for some of these disease states, you're going to run out of BIC by the time you're halfway through your commercial launch of your product. So the question is how do you maintain something that technically at a master cell bank well after the product is approved? So there's a lot of thought, obviously, in hemophilia A, which we have a partnership with Pfizer. We've spent a lot of time thinking about that, but that's the nature of the transient process. So your supply chain has to be thinking that much more. It's not just about the cell bank right now. Now it's the cell bank for the insect cell culture. It's the bank for the helper for your capsid. It's the bank for your transgene that goes inside the capsid. And that supply chain creates a lot of complexity. And for the long-term viability of these products, as we start to launch more and more of them, it's going to be challenging, very challenging to manage. And it also makes it very hard to increase, as we want to increase yield. One of the things in monoclonal antibodies that we're used to doing is focused on increasing cell mass. And if you get the cell mass up, you increase productivity. Well, that works for producer cells, but it's not necessarily a guarantee for the transient transfection process. And we've done some work where we have seen it requires a little more effort. It's a balancing act. So we are focused, obviously, on making transient transfection better. There's development work associated with that. But if you want to look at the technology solution, obviously it's moving towards this producer line, which will likely be in a mammalian cell system, not an insect cell. And it would require a yield hit probably, but over time, as we have done with CHO, and you see that development over the course of the CHO development from the early 90s and 80s into 2000, eventually 2010, we saw a Moore's law-like phenomena with yield, and that just took time. But I think with what we've learned from those with CHO, we should be able to accelerate. Once we can get this into a producer cell line, we can accelerate. There are a lot of conventional technologies for AAV and chromatography. The downstream is very similar, but we need to continue to innovate the right resins. We don't have that fully flushed out. And I think there's going to continue to be an investment there. But those are probably, I'd say, some, at least on the AAV side, where we need to be focused to get us out of the Wild West into a more monoclonal antibody type system where we see the productivity that we're needed. And the systems, we were highlighting 293 will work, even adherent work for these rare diseases. Like for Fabry, we're probably okay with what we have now. But if you want to look to, say, CNS, that's where we have 200,000 patients, and we have to deliver AAV at that scale. The other thing we're going to need to start thinking about is, well, maybe we can't operate in 2,000 liter scale. We'll need to be scaling these up, and we don't know yet what the scalability, particularly of a transfection process, would be. So there's going to be a lot of effort there needed, particularly as we want these genetic medicines to reach a broader patient population. And we're thinking through those manufacturability issues. I think, Doug, those are some of the key points I'd make.
Yeah.
And,
yeah, it's great. Wow. Yeah. You highlighted a lot of challenges there. And I agree with you. I think the idea of a stable producer line that can make AAV vectors like you can make recombinant proteins in CHO, that makes a lot of sense. I think I'm a longtime CHO cell culture process development guy, so I see the advantages of that. Thanks a lot, Andy. Daniela, how do you think about in vivo gene therapy space? And is your company interested in any certain types of technology angles in that space to invest in and to help move those products forward?
Yeah, absolutely, Doug. And I think just to echo Tim and Andy's comments, really agree and in line with a lot of what they mentioned. I think for us, we certainly see a big opportunity to disrupt a bit the status quo across both gene and cell therapy. I think one area is cell-free DNA manufacture. I think Tim mentioned the plasmid supply, and certainly a huge headache for a lot of the folks that we speak to. We speak to a lot of thought leaders in the space, and we partner with many biopharma, small biotechs, and companies that are innovating therapeutics in the sector, and that's definitely a headache that they're looking to solve. I think the other area is really and it goes to more the other, but is non-viral delivery methods. We are, I think viral vector is one of the biggest drivers of cost and inefficiency in the processes. And because it is highly customized, it accounts for a significant portion of development costs. So when we think about that, we think that directly links to cost of the therapeutic and patient access ultimately. So we're looking at non-viral methods for cargo loading. Currently, those are present in less than 10% of gene therapy trials. There has been a bit of an uptick, but we believe that they have a lot of promise. And it's a matter of time until we see something, a technology that will deliver the efficiency, be able to deliver genetic material at greater biosafety, lower costs, and simplify manufacturing overall. So waiting for that technology to appear for us.
Yeah, I agree. Viral vector manufacturing, whether it's lentiviral vectors or adeno-associated viral vectors, is somewhat challenging and complicated, both in terms of yields, as we've been talking about, as well as consistency from batch to batch. And yeah, I think a lot of companies are very interested in non-viral mediated gene transfer. When do you think that's going to be a reality for us?
We've
replacing AAV, for example.
Yeah, exactly. Well, we've been really active in the space. We spent several years canvassing the landscape and looking at different technologies. It led us to ultimately make an investment in Selfie Biotech, which is San Carlos based, microfluidic based approach to a virus-free cargo loading, and they're able to do this basically by microfluidic constrictions and allowing those cells to then pick up the cargo that's in the fluid surrounding. So this is a group that has, we're very proud to say, they've got several Nimble grants, several proof of concepts underway with major groups, and hoping that's one of the technologies that prevails. Obviously, there's a lot of other areas and a lot of other approaches out there, whether it's nucleic acid or currently electroporation is a pretty standard approach, but all of which have challenges with cell viability and making sure that you've got a safe product at the end of the day and high viability of cells.
Yeah. Thanks for those comments. Yeah, appreciate that. I'm going to turn now to ex vivo gene therapy products, also known as gene-modified cell therapy products. And Orchard Therapeutics is working on a number of these, as well as Sangoma. So I'd like to pose the similar question that we've just been discussing with in vivo gene therapy products to Ran, to comment on what do you see as the key manufacturing challenges in the autologous ex vivo gene therapy space, and where do you see technological solutions, sort of state of the art being employed now to develop and manufacture these products, and where do you see technology needs or future technologies that can really help move the needle, so to speak, and make the development of these products and manufacturing easier?
Yeah, thank you, Doug. Good questions. For autologous ex vivo gene-modified cell therapy products, I really see three significant challenges in the development and the manufacturing of those products. So the first one, I have to say, is to generate the product and process understanding. For example, we need to understand how the product quality attributes affect the clinical outcomes. Gene therapy is still in the early stage, and I don't think that we have a decent, thorough understanding in that aspect of that. That creates a challenge, in terms of how we control our processes so that we can deliver consistent product quality. So that related to the second one is the control strategy we put in place for process and the product consistency. And obviously the third one, many of the panelists already touched on, is around the manufacturing efficiency to reduce the production cost. And I also have to say that, one thing is also the cycle times for production, QC, and product release, so that we can get the products to patient faster. So from the technology perspective, maybe I'll just touch on the analytical piece first. There are many analytical technologies exist for product characterization to improve and enhance our product and process understanding, such as qPCR, ddPCR. We use those technologies for our product characterization, flow cytometry, CE for potency, and the cell characterization. And those technologies have provided insight for products' quality attributes. I think the challenges are actually connecting the dots with the clinical outcomes. So perhaps that the industry and academics, of course, need to take a slightly different approach to look at multi-omics and try to connect those dots. From the technology solution perspective, I think I would agree with Tim. There are a lot of progress made, but we still have many assays methods are quite behind compared to the biologics, and there are room to improve to make those methods more accurate and more robust. And perhaps multi-attribute assay could also add potential value to streamline the product analysis. Now, for the control strategy, I think the question, especially for Orchard and our company is autologous ex vivo gene therapy company, and we make one batch for one patient. So what we are facing is the variability of the starting materials from patient. And I think that's probably also applied to the allogenic companies that, they will see the variability from healthy donors material as well. So the question is that, would the traditional approach an effective way to deliver consistent product quality, or should we consider adaptive control strategy, meaning adjust the process parameters for individual batch based on our understanding of how process would affect the product quality attributes? We're still in the early stage to explore that. I'll touch on a little bit about the manufacturing efficiency. I think Andy and the team comment on those aspects already. So we use the lentiviral vector for our products. The current process is using transient expression with adherent culture. We have one process in suspension culture as well, but in general has a very low recovery in downstream process. We are in the process to develop stable producer cell line and using a suspension culture with more effective purification process. And we believe the new process will improve the yield and make the process more scalable. For the cell product, for HSC based therapy, one of the challenges is to introduce gene to the stem cells. It's a little bit different from the T cells. So we put quite a significant effort to improve transduction efficiency, from identify novel transduction enhancers to optimizing the transduction conditions. We believe that's a key for our products to reduce the quantity required for the vectors. Now, the cell process itself at the current stage, there are technologies to close certain steps of the processes and also make the process more automated. I think there are still room to improve and to make the process fully closed, functionally closed, and fully automated to reduce the labor content and the manufacturing cost. So I also touched on a little bit about the cycle time for product release. I think we have made quite progress in mycoplasma with PCR method and rapid sterility testing that is also required for the cell products. But many of the release testings are still cell-based assays and are quite labor intensive, take much longer to complete. That make the cycle time pretty lengthy, especially for some of the viral vector testing. So I think automated and rapid method for product release would be very advantageous.
Great. Yeah, all of those things ring true to me. Having spent time at Juno Therapeutics, working on CAR T cell therapy, I think you touched on a lot of things that definitely the CAR T cell companies are facing as well. Yeah, thanks, Ran. That was great. Andy, I'd like to ask you the same question. And one thing we haven't touched on too much is I know Sangoma is working on both autologous as well as allogeneic gene-modified cell therapy products and using your zinc finger technology to support manufacturing of the allogeneic. What do you see as, you know, the time frame and the challenges of moving to allogeneic? And yeah, please comment on any other sort of things you see in the you know, ex vivo gene therapy arena that you see as the biggest challenges that really need some technological solutions.
Yeah, no, and cell therapy is, you know, very new to me. While I know viral vectors very well, worked with them my first 10 years at Merck. It was my first job. I developed a Hep A vaccine, Hep B vaccine, worked on Gardasil, HPV a little bit, helped build a clinical facility and make all those products. So I understood these complex viral vector type products early on in my career. So AAV was not, you know, for me, didn't seem as big of a hill to climb. And then obviously I had purview on these are the things we need to address. Cell therapy is obviously new to a lot of people. In my 2 years at Sangoma, I've gotten to learn a lot about it through our work on with a CD34 based process for treating beta thal and sickle cell in a partnership with Sanofi. And then this deal with Kite that I'd mentioned to develop an allogeneic CAR T. So we hope to have that in the clinic at the end of this year to understand where that goes. But I will tell you that Allogene has reported recently using the same approach. They use talons. We're going to use zinc fingers, or use our zinc fingers to modify the same portions of the track and the HLA, the various genetic indicators and receptors that modulate graft versus host and identify a cell as itself, which in the end causes problems with when it's recognized as foreign to the patient's immune system. So using our technology, just like Allogene used talons, we're going to use zinc fingers and we're going to modify those, and we should be able to see the cells respond as kind of basically inert cells that carry the necessary genetic material to do whatever steps are needed to treat the disease. And that is something that I think is happening now. I think that technology is very likely to start taking hold, and Allogene has presented results. And you guys, if anyone bought Allergen Stock before that, probably doing really well right now because the data was compelling enough that got people excited. And this is an exciting development. Now, one of the things I will point out is that their starting material is healthy donors. So that basically means if you're going to do allogeneic cell therapy, you're going to be in the blood banking business, right? You're going to be harvesting cells routinely from healthy patients to use as starting material. You'll do the genetic, you'll purify them to get the T cell you want. You're going to then genetically modify it using probably electroporation or other similar technology to insert the talons, or for us, the zinc fingers, and a CAR T construct. And that will then allow these cells to behave allogeneic. But the problem is you're starting with patient's blood. Now, one of the key developments to scaling this up, and the problem is if you even do this, it's not going to be, you're not going to be able to use it. For example, we've been looking at it for HIV. We've had an active HIV program for some time. It's one of the first things we looked at. And that is going to be impossible to do autologously with the numbers, and even with healthy donor, where you can get 100 patients per healthy donor batch. I think that's what Allogene has projected, and that's actually what we think we should be able to get. Now, obviously, with improvement in technology and expansion of those cells, we might be able to see more. But even that, the limitation still is you're starting with a human blood source, which everybody knows the problems with that. Now, we could start earlier, and you're going to need to start with human cells somewhere, and that's where we're looking at iPSCs. And that's, I think, where a lot of the industry is now looking, that is the starting material for whatever cell you want to make. You'll generate banks of iPSCs, and you're going to edit those iPSCs to do whatever it is genetically you want them to do, and then you will differentiate them into the cell type that you want, be it T cells, be it NK cells, or for us, we have a platform in Tregs that we're looking at. This is still probably 3 to 5 years off. It is, so we're relying on healthy donor until we get it to work. But that is the game changer. It'll allow you to actually establish a bank. So you'll start with a human cell at some point, just like, you know, we've done with a number of our, like the HeLa cell line and HEK293. It all started with a human, one human patient. And then from there, though, you can expand and you won't have to keep tapping into patients. So I think that's an important development to make allogeneic cell therapy really a traditionally off-the-shelf, patient accessible for broader diseases. Otherwise, it's going to be mainly focused on, you know, like maybe 1,000 to 10,000 patients, you know, max per year type treatments. I will say, and both Tim and Ron had talked about analytics, and it's something I didn't maybe speak enough about it. I didn't mention it with AAV, but I want to reinforce it for both of these programs because we have, you know, process and analytics is the number one pillar of our manufacturing strategy, is getting the right process and the right analytics. And as you guys all know, you don't have a process if you don't have the assays to assess whether that process is any good. And that really has to be a focal point. And I just want to say a few words about AAV, then I'll get back to cell therapy. But going back to analytics, there's some basic stuff. Like I said, we're still in the Wild West. We don't even really have a good dose estimating AAV assay. Using qPCR is plus or minus 15%. Do you know what the FDA expects? If you want to put a certain amount of AAV in the bottle and say what it is, you have to be plus or minus 5 to 10%. That's the expectation. If you're going to have a, you know, this is your, how much drug, it's your dose, right? And it's defined by a certain amount of material. Well, you need to be able to measure that reliably. So we're looking at ddPCR to get that. Now, I don't know if that solves that, but that's one of those very basic manufacturability issues that we haven't really actually fully addressed. And it's something that we need to continue to evolve. Analytics is a priority here, and we'll need to be spending a lot of time. So that's just one of a couple I would highlight. Yeah. Getting back to cell therapy. Getting back to cell therapy, and then I'll wrap up, Doug. We talked about the critical quality attributes for cell therapy processes, and part of the issue is the clinicians actually don't know 100% what it is that's important for efficacy yet for some of the things like Tregs, which is something that we're trying to establish clinically. We think we have a pretty good idea, and we have a number of assays, so we're basically looking at it very broadly. For CD34, getting back to one that we know, long-term progenitor cell population, which is something that we initially didn't realize was important, but we found out through our clinical trial, we have a lot of cells. We have good indels, which we can measure. You know, we can measure the percent of genetic modification. It was high. And we were pretty confident, but why wasn't it manifesting itself clinically like we wanted? Well, we found out, hey, you know, we hadn't been looking at this, and now we are. And now we're modifying the process to make it better. As you can see, without good analytics, you're dead in the water. And in the end, some of those analytics, we need our clinical team be telling us, hey, these are the things we need to measure and optimize. So this is a cool thing. And actually, it's a crazy thing about cell therapy. It is a very close relationship that I've never had before with clinical, right? We are talking to them daily about how we have to run and optimize this process. So it is way different than the CHO from monoclonal antibodies. They just said, give me the monoclonal antibody. This is, you know, high level what we need, and that's what we focus on, making as much with a certain amount of purity. That is not the case. So it's a very interactive with clinical and certainly with regulatory, because we're trying to figure out how to navigate that too. So it's like I said, a bit of the Wild West, we need attainment, but it's exciting.
Yeah.
And
Yeah,
I'll
that's
make one
great.
last point, and then I'm done. Manufacturing, ideally, if we want to get this to where we want it to be, it can't be exciting, right? It needs to be routine. It needs to be, you know what you need, you know what you need to make. And that's what we got to do. We got to get out of the Wild West and get this into something a little less exciting.
Yep.
And analytics
Sounds like
is the key to doing that.
Yep. Sounds like something a CEO would tell their head of technical operations. Quit making it so exciting in manufacturing.
Yeah, boring is good in manufacturing.
Yep. I want to turn back to Tim and change gears a little bit here. The CDMO space is very, very critical to a lot of companies, especially startup companies trying to develop their products. And some CDMOs seem to offer more of a technology platform than others in terms of what they provide clients that come to them. How do you, through your partner ViralGen, how do you think about the importance of providing, technology platform to your customers versus, just working with the customer's technology and trying to do what the customers ask? How do you view that from the point of view of ViralGen?
Yeah, thanks, Doug. So, ViralGen's maybe a little bit of a unique kind of circumstance because, we are really a true platform CDMO for AAV. For example, I guess comparing it to conventional, monoclonal antibody sort of CDMO business, I don't think you can really expect to just buy capacity in the gene and cell therapy space when you're looking for a CDMO partner. It's not enough to just have the bioreactors or the prodigies or the cell stacks or whatever the case may be. You really do need capability and expertise. And I think the advantage of the platform is that gives your partner the opportunity to really become expert. If you're running, fundamentally the same process, sort of day in and day out for every construct that comes in, you really develop that expertise and that ability to troubleshoot and to continue to optimize. You have the chance to continue to implement improvements to that platform. So for example, going to higher density cells, as some of the other panelists were describing, or replacing ultracentrifugation with chromatography processes and still optimizing your full to empty capsid ratios. Trying to do that on a process by process basis, with a client material coming in that looks different, I think is really challenging. So again, at ViralGen, we're really, the trade-off is that it's a little bit of a one-size-fits-all, right? We're AAV, it's focused entirely on AAV. It's using the Askbio technology platform, the Pro-10 cells, and the manufacturing process. So, customer has to sort of buy into that. But I think what you get then is that sort of consistency, that expertise in the process. The way that we typically engage with a customer at ViralGen, we'll do a 2-liter feasibility batch. And within 2 weeks, generally, we've got some small research quantities of AAV particles to send back to the client for them to do potency and other assays on. And if that looks good, then as long as we have plasmid, we can go essentially directly into clinical production. And again, plasmid is a rate limiting step today, as we've touched on, a couple of us have touched on. And again, that's where some of these, again, Daniela was mentioning the cell-free. Again, our JV for Touchlight AAV is just that. It's sort of enzymatic synthesis of these linear double-stranded covalently closed DNA fragments that we know that we can effectively transfect and generate AAV particles just as well or better than we can with conventional plasmids. So I think that those, ultimately then being able to go directly, into clinical manufacturing really circumvent process development, at least for early clinical. You get material into the clinic quickly and with high productivity. We're able to sort of consistently see 2 to 3 E17 purified viral genomes per 250-liter batch for the most commonly used serotypes like AAV8 and AAV9. We have a DMF available. We basically have your CMC section already written, to a large extent by virtue of the ViralGen DMF. So that's a big advantage for customers, seeking to get into the space. I think where customers tend to have some heartburn about platform technologies is in IP control and licensing and so forth. And I think that's another kind of unique thing about Viralgen. You can, by signing a fee-for-service contract at Viralgen, you have access to this Askbio technology platform. There's no licensing, there's no royalties, there's no encumbrance to that. The trade-off there is that we own it and we retain it. So again, it's there's a little bit less flexibility in terms of tech transfer and so forth as compared to conventional CDMOs. But we believe that that's a trade-off that makes a lot of sense. Kind of given what we think we can offer in the space.
Great. Yeah. Thanks, Tim. I'd like to ask you, Ran, if you have any thoughts too on I think you guys use CDMOs for producing some of your products and vectors. How do you consider how do you choose a CDMO in terms of about technology and what their offerings are in the technology space?
Yeah, thanks, Doug. I think it's very important that the CDMO have the technology capabilities to offer, and which could help reduce the development timelines, improve manufacturing operations, and enhance product and process understanding, especially for the product companies that have not yet built internal capabilities. The technological offering could be crucial to move the product development forward, allowing the product companies to fly the plane while building it. So I really think it's a very important feature for the CDMOs, not just to have the capacity to make the product, but also have the technical know-how to develop and improve the processes. And even for the established product companies, and I think CDMO technological offers could potentially augment the internal capabilities. So for Orchard, when we first started, we didn't really have any process development capabilities, and we only built that capability over the past couple of years. So it became very important for us to work with the CDMO that have that capabilities first. And even now we have our internal capabilities. What I found is that the good process and analytical technology platforms developed by the CDMOs very often leveraging the understanding across different products. That could be very beneficial. So I think overall, I would say when you select CDMOs, obviously the robust quality system and the manufacturing capabilities are key to consider them as a manufacturer of your products, but the technological capabilities is very, very important. It's critical.
Great. Yeah, thanks for that. I think we have just a little bit more time, so there's just one more question I wanted to ask Daniela. You have sort of a unique point of view, potentially as being part of a venture capital investment company, investing in new technologies or companies that have interesting technologies. How do you go about really evaluating the promise or potential of new technologies in the cell and gene therapy space? I mean, do you have any important ways, metrics, ways to evaluate? How do you really know how to what are the key technologies that you'd like to invest in?
Yeah, absolutely, Doug. Thank you. So maybe going back a bit, certainly we've identified in the cell and gene therapy landscape several key pain points across that landscape. It's all of the aforementioned content that the team here has mentioned. But just what leaders tell us as their first priorities often drives what we're interested in terms of looking at novel tech. So some of them are super interested in closing the process and the interconnectivity between unit operations. Some are focused very much on non-viral delivery methods. Some are looking at upfront cell selection and cell characterization. Others are downstream assays because it's such a labor-intensive process. So there's a whole range of sort of on the technical side, specifics within this landscape that drive us to look at those things. I think what's really interesting is that dynamic is a great go-between. We've found that basically bridging, as an investor, bridging biotech and biopharma and life science entrepreneurs, that we can translate those needs of the therapeutics innovators to the technology innovators, and also help a bit with setting expectations of the therapeutic innovators, from everything to GMP ready, automation, documentation, DMFs, and supply chain, right? So there's a lot that's wrapped up in that. I think because our team has uniquely sort of commercialized novel technology and manufacturing approaches, as well as CDMO services, we have the expertise, the experience, the network, and the knowledge to help these entrepreneurs to do that. More specifically, if we think about, you mentioned sort of what do we look for in these opportunities? I think key for us really is proof of concept. Utility, is there a demand from the marketplace? So, for example, we work really closely with industry leaders. They certainly help in terms of those assessments, those technical assessments, in terms of statements of work or scopes of work that they might set up with innovator companies. But if there is some kind of customer engagement at that level, then it helps us be more comfortable with the technology. We may even contract another group to help support that bench work to validate the results that they're receiving. But I think one important aspect here is that the reality in this space, there are systems in place today that are far from ideal. They're not fully industrialized, but they're used maybe as a carryover from bioprocessing and traditional biologics, and they may not be the best solution. I think a great example is, for example, the wave bioreactor. When I think back to our launch and how that evolved over time to become sort of a platform standard in terms of cell therapy and patient-specific cell therapies, that's been a really interesting story. More specifically, I think now, compared to then, customers at the time were really sort of willing to take a chance on novel technology. I think it was simple, it reduced labor, it worked, and though it may not have been fully baked in 1999, people started implementing it and speccing it in, designing it into their clinical trials. I think that was very lucky for us. And I expect today that bar is much higher. And the expectation, I think customers and therapeutics innovators expect a more mature technology before designing that into their process, because the risk is so high. At the same time, we're talking about very precious material across the board, and those companies might not be willing to take a chance. On the flip side, the pain points are so great that I do think there are some intrepid groups that are making the leap, helping those smaller companies along. But those life science innovators really need to put forward something that is robust, that has the supply chain in check, and that not only is robust, that it looks robust, that it really walks the walk.
I remember when the wave buyout and thought it was kind of silly, but it turned out to not be silly, and it's used everywhere, and I've used it myself everywhere, and it's gone on to not just help monoclonal antibody production, but as you said, cell therapy. It's a workhorse, and so it's a very exciting or very interesting story, the wave bioreactor. So thanks Thanks for that. Well, I think we're about out of time. Unfortunately, this has been a very excellent, interesting, entertaining, and informative discussion. I want to thank each of you for participating, and yeah, I think it's been really informative. So, thank you all.
Thank you, Doug.
Thank you, Doug.
And yeah.